Hello, everyone. Welcome to another Nerd. For those of you joining us for the first time, we are the neural engineering research venture, and our aim is to bring neuroscience to life in Africa and around the world. And who are we? We consist of myself, Julianne, and Siobhan, who will be your hosts for the evening, and Bobby, who you can always find lurking in the comments. Then we have a couple of announcements. The first we have here is the Deep Learning in Daba mentorship program aims to strengthen the African machine learning community via the development of fundamental skills. Computational neuroscience is included in the scope of these sessions. The program matches members of the African AI community with mentors for short-term personalized interactions across a wide range of topic areas. You are encouraged to either join the excellent global network of mentors, or if you identify as part of the African machine learning community, please do sign up as a mentee. And Siobhan will be sharing all of these links in the comments section. Then we also have the announcement for the DeepMind scholarships at Stellenbosch University. The applications are now open, and these are open to African machine learning community members looking to pursue a MSc or a PhD in South Africa, starting in January 2022. The scholarships cover university fees, health insurance, accommodation, living expenses, and opportunities for international conference attendance. The recipients will also benefit from mentorship from the DeepMind researchers, and the application closing date is the 31st of July, 2021. Then we have the Neuromatch Academy 2021 that will take place over the next 2 months, starting with the Computational Neuroscience Summer School from the 5th to the 23rd of July. This will be followed by the Deep Learning Summer School. Applications for the interactive track are already closed. However, you will be able to find all the content online during these dates. You can follow along in real time via the Observer track. So be sure to check out all these opportunities via the links provided. Then also, these are great links to save in your browser. Worldwide Neuro and the ABC Meetup regularly host online events across an amazing spectrum of neuroscience. And Worldwide Neuro now has expanded to include the larger fields of worldwide science. They are an amazing platform for anyone looking for a job or looking to promote themselves, so your next job can find you. Okay, then as always, we have a couple of house rules. Please do use respectful and inclusive language on our platform. Then also make use of the Ask a Question feature throughout our speaker's presentation. So if you post your questions there, we will be sure to get to them in the question and answer session after the presentation. You're also welcome to post any quick comments or questions in the comment section. We will be monitoring that section. Then remember to vote for your favorite questions in the Ask a Question section. After the presentation, we will invite you on screen to ask any of your questions live. If you do not want to come on screen, or if for some reason your internet connection is not stable or you don't have a mic, just please add "please ask" to your question, and then we will ask on your behalf. And if your connection is slow, try to refresh your browser. Alternatively, you can select the compatibility mode from the audio-visual help button at the bottom of your screen. And if your connection fails completely, don't worry, the talk will be available to stream after the event. I will now hand over to Siobhan, who will be introducing our speaker.
Thank you, Julianne. I'm really excited to introduce Mike this evening. Mike's research has two primary focus areas, the first being the investigation into neural circuitry underlying midfrontal cortical theta oscillations. This is a rhythm linked to cognitive control and behavioral adaptation in multiple species. The second focus is the development and evaluation of data analysis tools and algorithms for uncovering structure and dynamics in large-scale multivariate neural data sets. I would also like to take this opportunity to personally recommend Mike's amazing collection of learning resources, which can be found both online and in print. These cover everything from time series analysis to signal processing, as well as getting into the finer details of statistics and linear algebra. There are courses on Udemy, which I would recommend, but a lot of the content is available on YouTube if you're looking for help on a specific topic. One of my favorites being the series on the Fourier transform. Thank you very much for joining us this evening, and we look forward to your presentation, Mike.
Great, thanks. So let me see, share. Thank you, Siobhan. And Juliana, and I know there's always a lot of people who are involved in inviting and guest speakers and organizing these things. So thanks to everyone who organized this, and thanks to all the people who are joining and participating. You can see my screen, I guess, right?
Yes, we can.
Okay, great.
Thanks.
Yeah, so my talk is broken up into 3 sections. I'm going to say a little bit, so spend maybe half the talk on like parts 1 and 2. That's a little bit about the research that that we're working on in my research group, and how we got there. And then the third part will be on multivariate data analyses. So let's start with some ancient history, depending on how old you are. You know, this may or may not feel ancient, but it's going back to like 2004, I guess, just for a little bit of context. So, you know, everyone makes mistakes. We all make mistakes. I hope you don't feel that spending an hour with me here is a mistake. But mistakes are very human to make, but not only humans. Animals make mistakes. You see mistakes all over biology. In fact, there's even an argument to be made that at the most fundamental, lowest level of biology, mistakes are just intrinsic to the structure of life itself. And I'm thinking in particular about random mutations in DNA, without which we wouldn't have, you know, we wouldn't be here. So I think there really is something special and universal about mistakes and learning from mistakes and making mistakes in biology. So, but of course, you know, we also don't like to make mistakes. We try to avoid making mistakes. We try to continuously monitor our environment and also monitor our actions in case we are about to make a mistake, and then engage these very rapid corrective behaviors to prevent making that mistake. And so, so this is something that I was interested in when I started doing cognitive neuroscience research. And at that time, you know, it was known that there was this specific EEG time domain potential called the ERN, the error-related negativity, and another potential called the N2. But I was interested in taking a slightly more physiological perspective, which for scalp EEG, I think looking at time-limited and frequency band-limited oscillations is a good way to try to link this non-invasive measurement that we get to underlying physiology. So at that time, like around 2004-ish, there was really very, very little known about the time frequency characteristics, the electrophysiological characteristics of almost making a mistake. So this is something that I and my colleagues set out to figure out. So what we do is cognitive tasks with human research participants where we have them do tasks that elicit what we call response conflict. And response conflict is when you experience this competition between an automatic response and a top-down response that is instructed through the task. So this is typified by something like this, which comes from a task called the Stroop task, which you might have heard of. Here, the goal is to say the word that the color, the color that the word is printed in. Excuse me. So here you would say the word green. You can see I'm already tripping over my words because you have this automatic tendency to say the word blue, but instead you have to override that and say green. So you're likely to make mistakes, which I just did. And so I experienced this frontal theta thing. So we look in particular at the EEG trials during the EEG activity during correct trials where the subjects were likely to make a mistake, like this kind of trial where they actually did say green, but you know they were slower to respond and they're more likely to make mistakes. Now, when you look at the EEG signal, you see this kind of ongoing wobbles, and then after the stimulus onset, and sometime around when they press the button, you see this increase in the amplitude of this ongoing rhythmic activity. And this rhythmic activity is around 6 hertz, so we call it theta. And if you look at how this signature of almost making a mistake, of response conflict, is distributed across the scalp, then you see it looks something like this. So this is just the one electrode where it's maximum, but we record this with anywhere between 64 and 256 electrodes, and the topography is very consistent. So it's kind of a funny thing because, you know, when you make a mistake and you screw up, and, you know, what's the first thing you do? You know, you say, oh shit, like, you know, you whack yourself in the forehead, which turns out to be pretty much exactly the location where we see this effect maximum. Pretty sure that's just a coincidence, but it is sort of fun to think about. Anyway, we call this feature of the data midfrontal theta because it has a midfrontal topography and it is in the theta band. Now, of course, this is just one single trial of data, and it's filtered, it's temporally filtered at the frequency that I want you to pay attention to. So this is not super compelling scientific evidence, but we've done many of these experiments over over a couple of decades now. Let's 15 years. We've done many of these experiments with many different kinds of tasks, many different ways of looking at the data, and many different kinds of analyses. And what we see when you open up the analyses to look through, you know, all time points and all frequencies and all electrodes, you see that when you average the conditions together, so just looking at the cortical responses to the task in general, there's a ton of stuff that we see. So there's frontal theta that you see here around 6 hertz. There's some beta suppression, which is related to the motor response. There's all sorts of dynamics happening at the back of the head related to processing the visual stimulus and so on. So lots of stuff happening in the brain during this task. But when you specifically make this contrast between trials that contain response conflict and trials that don't, so that would be, you know, this stimulus versus where the word blue is actually printed in blue, then basically all of this other stuff just completely disappears. It totally drops out. And then the only thing that remains is this fairly transient, so fairly brief, fairly band-limited response in the theta band, and it's also pretty topographically specific to midfrontal regions. Sometimes this particular experiment was involved with spatial manipulation, so we got some parietal theta in here as well. But generally, it's really just remarkably specific. This is a very idiosyncratic electrophysiological signature of response conflict. So yeah, so we spent a lot of time. I could give, you know, like a 2-hour talk just about all the details of all of these findings, which I won't really trouble you with too much. But if you're interested in having, you know, what was sort of my thoughts about the overview of the field at that time, you know, this paper is now 7 years old, but you could look in TINS in 2014. One of the things that I wrote here is, you know, I was thinking a lot about the specificity of midfrontal theta and whether midfrontal theta really tells us something about response conflict per se, or whether it's just a generic signature of activation of the prefrontal cortex. Because there's many other cognitive tasks that also are associated with theta oscillations in the prefrontal cortex. So it does seem like many different kinds of computations in the prefrontal cortex are manifest as theta EEG activity. But, and this would be part of the longer version of this talk, but when you look specifically at this response conflict dynamics, you see that this pattern of, you know, it being this transient and this specific to frequency and these electrodes, you really only see this pattern whenever you look at response conflict, and you don't see that idiosyncratic signature when you look at like memory or reward feedback or, you know, other kinds of processes that also elicit theta. So the data led me to believe that there is one very specialized, specific microcircuit in the medial frontal cortex that produces theta oscillations that also generates response, or sort of detects and signals response conflict. So it was a very strong prediction here in this paper that there was basically just one unified, you know, one unitary process in the brain for response conflict. I'm stressing this because this comes to be, I'm going to come back to this point later in the talk. So it's somewhere around like 2013 or 2014 or something. I had a slight mental meltdown might be a little bit exaggerated, but I did, you know, I had a moment where, you know, I woke up one morning and I saw the possibility of spending the next 35 years of my career just doing the same EEG experiment over and over and over again, you know, with like slightly different variations. So, you know, let's see if it works with purple stimuli. Let's see if it works with sounds instead of vision. And I don't know, it filled me with terror. And I thought, man, I have to do something else with my career. I can't just study this one single thing for the rest of my life. So then I was thinking about what else could I do? I was still interested in this midfrontal theta dynamics, but so there were many possibilities that I thought about. The role of theta in other cognitive processes, midfrontal theta and brain diseases, trying to bring our findings from the lab, you know, sort of artificial lab experiments more into real world situations. All of these, I think, are important avenues of research, and I'm happy that there are other research groups that are investigating these topics. But this is the one that really stuck to me because, yeah, I think in the spectrum of cognitive neuroscience, I'm much more interested in the neuroscience than in the cognitive part. So midfrontal theta and underlying neural circuitry. And the question here is, you know, we measure this EEG signal, this non-invasive signal, which is great. I'm a huge fan of EEG. I think we can do even more with EEG than what the field currently does. But on the other hand, you know, it's a very aggregated, indirect signature of brain computations. And the thing is, you know, cognition doesn't come from these wobbly lines that we measure with EEG. These wobbly lines are, you know, they're coming from the brain and they're meaningful. But this is not what we really care about as neuroscientists. What we really care about is what are the underlying computations? What are the computations and configurations of neural circuitry in different types of cells distributed across different cortical layers and interacting with different cortical fields and modulations by neurochemicals and so on. These are the things that we're really interested in, but this is what we can measure. So that's a big gap there. So what I wanted to do is see if we can understand something about the underlying neural circuitry based on what we can measure with EEG. So the question is, you know, basically something like this. Can we use this macroscopic signal to tell us something about the underlying neural circuitry? So it's a very simple question, but it turns out to be just really fucking hard to do. It's hard theoretically because of the complexity that's happening at this spatial scale. It's hard technologically, it's hard conceptually, it's hard in terms of data analysis, it's hard in terms of just even collecting the data in the first place. It's just a really hard problem. But I had like a lapse of sanity, and I thought, let's just go for it anyway and see what happens. So all of this was happening like in 2013, 2014. I wrote a couple of grants all about this topic, and fortunately they were funded, which was, as much as I had a lapse of sanity to even think that I should do this, I think the ERC grant committee panel had a lapse of sanity in thinking that I should get this money. But anyway, they gave me the money. So that leads to part 2, which I call the messy parts, because if any of you in the audience do lab work or anything with biology, empirical biology biology in general is just really messy. It's dirty business. So this all started in 2005 when I moved from the University of Amsterdam to where I am now, which is the Donders Institute. And so here's the main empirical goal of my lab, which is to understand something about the biological origin of midfrontal theta. And we do this by manipulating and measuring microcircuit components. And this is not something that's really possible to do in humans. So therefore, we had to move to animal models and build an animal research lab using mice and rats and invasive electrophysiology and optogenetics to try to investigate these microcircuit components. So a lot of this research is still ongoing. and it's still kind of new. So I don't have a lot of like really strong conclusions to draw. So if you were hoping that my next slide was going to be the answer to this question, then I'm sorry to disappoint you. I also want to make sure that I have some time for the last part of the talk, which is I think what was advertised here. So I'm going to go through this rather briskly, but my main goal here is just to give you a bit of a sense of the kinds of experiments that we're running, the kind of research that we are doing in the lab. So this is a project with looking at the role of phasic dopamine and synchronization, because that's thought to be important for normal cognition and also disrupted in several brain diseases ranging from Parkinson's to schizophrenia. So we can optogenetically up and down regulate the dopamine cells in the VTA that project to the striatum and or to the prefrontal cortex, so we can drive phasic stimulation in the of VTA, ascending VTA neurons, while recording electrophysiological activity from lots of electrodes, up to 200 electrodes in the prefrontal cortex and in the striatum and VTA, and we also have a couple of EEG electrodes on the skull as well. Let's see, so yeah, we get some nice expression of the virus. And here's just a little bit of a taste. So here we are stimulating the ascending dopamine fibers from the VTA to the PFC at 4 hertz. And what you see is actually the response in the prefrontal cortex is around 9 hertz, and it's harmonics as well. And it also takes around half a second to build up. So it's pretty interesting because this is not just a response to the light stimulation. Instead, it's a response to the buildup of dopamine in the prefrontal cortex. And it's a nonlinear response, so it's responding at its own resonant frequency, not just the frequency that we are injecting into the VTA. So yeah, there's more to say about that. We also have some neat dual virus experiments that we're running in mice, where we can selectively modulate the excitatory inputs into prefrontal cortex and also the local interneurons inside the prefrontal cortex. And I guess one thing, so I just show you this experiment. So here's a little, this is pilot data, so we're still working on replicate or analyzing all the rest of the full data set. So here we're recording from the prefrontal cortex, and we give phasic stimulation to the excitatory, so the glutamatergic inputs from parietal cortex into the prefrontal cortex, and that elicits theta in the PFC. So these long-range cortical inputs elicit PFC theta, and then we can actually block that theta by combining this stimulation with inhibition of the somatostatin cells, which is a subclass of interneurons in the prefrontal cortex. So that's pretty neat. We get some causal evidence for the generation of theta in the prefrontal cortex. There's also there's ongoing work with more data. So we also have experiments, in particular in rats, where we have some behavioral tasks set up. So this would be like a Skinner box where the rat is in here, and there's nose poke holes and LED lights and little speakers, and we can deliver water rewards to them to train them to do particular tasks. In particular, we train them to do what's called a Simon task, which is conceptually similar to the Stroop task that I showed you in the very beginning. So we basically elicit response conflict in these rats. And we do this while we're recording from the medial prefrontal cortex. So now, one of the questions that we asked, this is Marit, one of my PhD students who did the data analysis here, is how many thetas are there in the prefrontal cortex? So how many theta generators are there? Remember, I said I made this very strong prediction a couple years ago that theta reflects the activation of one very specific dedicated microcircuit that responds to response conflict. So that's visually depicted here. So midfrontal theta would be unidimensional, and yeah. So then the more that component is active, the more conflict there is. So we looked at this in our multi-channel data because we were recorded from 30 channels in the rat prefrontal cortex. And then I'll get back to plots like this later. Essentially what we found is that, in fact, there are multiple components, multiple theta components in the rat prefrontal cortex that are responsive to response conflict, that are engaged by response conflict. So here you see from this is what from one animal we see these are different components that we can linearly separate using multivariate data analysis methods that I'm going to talk to you about in a few moments, where we find distinct spatial patterns of theta, so distinct circuits in the medial prefrontal cortex that are all responding in theta and also are modulated by response conflict. So that was a little surprising to me, but I was happy to see that. And then Marit and I were talking more about this, and we thought, well, maybe there are also multiple thetas in the human medial prefrontal cortex. You just have to look for it in the right way. And so we decided to do that in human data. And these were human data that were collected a couple years ago, where we had simultaneous EEG and MEG. So it was 270 sensor MEG and also 50-channel EEG simultaneously in the same subjects while they were doing a response conflict task. So we applied a multivariate data decomposition method, which is going to be the main point of the third part of the talk. So, and it's based on covariance matrices. So this is the covariance matrix for narrowband filtered data in the theta range, and this is the covariance matrix for the broadband data. So you see that there's these kind of two strong patterns here. This corresponds to the EEG channels, and these are all of the MEG channels. So, so then we applied this decomposition, and we find this is the eigenvalues, so the power ratios, and we find actually that there are many, many theta components that exceeded a statistical significance threshold. So there's lots and lots of theta components, linearly separable, independent theta components in the brain. Now, not all of these are midfrontal components. Some of these are also going to be occipital or parietal. Some of these might also be some artifacts, and coming from blink artifacts or EOG artifacts. So we filtered through all of these significant components, the statistically significant theta components, and looked for components where the EEG topography matched a midfrontal template, because this is what we expect conflict-related midfrontal theta to look like. And they also had to be modulated by conflict. They had to be non-redundant components, and so on. We had a couple of stringent thresholds to make sure that we're really identifying true theta components. And then here you see the results from 9 different subjects. And this N here corresponds to the number of statistically significant midfrontal theta components that were modulated by response conflict in the task. And different subjects had different dimensionalities, so different numbers of components, but you can see it's, the smallest was 3 and the largest was 11. So maybe this one just has like a lot of response conflict or something. But, the fact that this number is even greater than 1 already disproves something that I said earlier. So it seems like the picture actually looks like this. So midfrontal theta is actually not a unidimensional process in the brain. It's not a unidimensional computation. Instead, if we look at, so S would be for the sensor. So this would be the sensor space, and then there is some smaller dimensional subspace corresponding to midfrontal theta, and that's depicted by the blue box here. And then there's an even smaller subspace within that midfrontal subspace that is responsive to conflict. And so this is a multidimensional representation, which means that there are multiple linearly separable theta generators in the midfrontal cortex. And then we can identify these the different basis vectors for them. Now, exactly, why are there such individual differences and what do these basis vectors correspond to? We tried to look at this a little bit in the data to see if these generators or the different basis vectors correspond to different features of the task or something like that. That's yeah, it was not very clear from this one study. But what was very clear is that midfrontal theta is not a unidimensional single process. It's actually a multi-dimensional computation. So yeah, so then I made this very clear prediction, which is completely wrong. And that is how I got canceled by neuroscience literature. Just kidding about the canceling part. Actually, you know, this does, nobody really likes to be proven wrong. And I did get a burst of midfrontal theta when I, you know, realized that I was wrong all these years ago. But it actually, you know, it is a very nice feeling, I think, as a scientist to be proven definitively wrong, you know, to make a clear falsifiable hypothesis and then find out that it's wrong. Because yeah, that's really, science only really moves forward from proving yourself wrong or proving other people wrong or having someone else prove you wrong. So it's much better to make a clear, definite, falsifiable hypothesis and risk being wrong than just to sort of be very wishy-washy and not really come up with a good hypothesis, and then you're never really proven or disproven. So so yeah, make clear hypotheses and embrace and even welcome being proven wrong. It helps you sleep at night, I think. Okay, anyway, that's pretty much all I wanted to say about the sort of topical part of my research. As Siobhan mentioned in the beginning, there's kind of like two main themes in my research group. One is the empirical stuff, which I just told you about, and another one is the data analysis stuff. So let me now tell you a little bit about that. In particular, I'm going to focus on one specific multivariate analysis approach. So let me start by saying that the traditional sort of, yeah, I'll call it traditional. The traditional and most common way to analyze data in neuroscience is called univariate. And there the idea is that each measurement, so each electrode or each neuron or each fMRI voxel is treated as a separate measurement, and the analysis is run separately on each individual measurement unit. The idea of a multivariate approach is that you take each one of your individual electrodes or neurons or even, you know, questionnaire items, and rather than analyzing them individually, you figure out how to pool them together to say something about the patterns that are distributed across these different electrodes or neurons. So the overwhelming majority of neuroscience research takes a traditional, this is called mass univariate approach, when you're doing univariate analyses on lots and lots of data measurements. Now, it's not wrong. I would never argue that univariate analyses are wrong, but I do think we can question whether, you know, that sort of univariate approach that has dominated neuroscience is really the best way forward, and whether multivariate analyses can give us deeper insights. The only, you know, there's a couple of assumptions that depend on exactly the kind of multivariate approach you take, but the most important assumption is that neural coding is spatiotemporally distributed, which is something, you know, that neuroscience has generally agreed upon, with some exceptions. So, you know, we say that neural computations are distributed over some population or ensemble of neurons. And if this is true, if you take this assumption as true, then it means that measuring the data and analyzing the data from only one neuron or electrode or voxel is going to miss that pattern, because that pattern is actually distributed over multiple sites. Now, that doesn't mean that you'll completely lose the pattern, but it does mean that, you know, at best, you're going to have a reduced signal-to-noise ratio, so lower quality data. So that kind of advocates for a multivariate approach. Now, there's many multivariate approaches. There's decoding, which is sometimes called multivariate pattern analysis, principal components analysis, independent components analysis. You've probably heard of these analyses. Deep learning is also like the new kid on the block, which was also mentioned at the beginning, before I was introduced. Now, all of these methods are great. And, I could have a 2-hour version of this part of the talk where I go into more detail about these analyses and say, and discuss their advantages and limitations. But the idea is that all of these multivariate analyses require optimizing something. They're all based on some optimization. So the question is, what are you optimizing for? And I would argue that often for hypothesis-driven neuroscience research, these methods are not necessarily optimal. They're not necessarily the best because it's difficult to get really the right control over the optimization criteria. So what I often argue for, which is a really great mathematical background for lots of analyses, is something called generalized eigendecomposition or generalized eigenvalue decomposition. It's often also abbreviated just GED. And here, the idea is you optimize for a covariance signal-to-noise ratio or a covariance contrast. So you're going to take two covariance matrices and contrast them against each other and kind of pit them against each other. That's the idea of GED. So a covariance matrix is a square symmetric matrix that encodes all of the pairwise linear interactions across all of the data channels. It's basically like a correlation matrix, except it's in the same scale as the data, the same units as the data. Covariance matrices are really great because they are a compact representation of basically everything linear in the data. All the linear interactions and so on are all contained in the covariance matrix. So I'm going to introduce GED as an extension of PCA. There's multiple ways to introduce GED, but in this case, I'm going to start with PCA. So this expression here is the objective criteria. This is the optimization criteria of PCA. So the idea is you have some covariance matrix S, and you want to find some vector W, which is a vector of weights, so a way to weight all of the data channels such that the weighted combination of channels maximizes the energy or the power in S. And then there's a slight abuse of notation, so apologies to the mathematicians in the audience. But when you find the W, the set of weights that maximizes this ratio, then this ratio just reduces to a single number, and that we call lambda. Now, PCA is not really suitable for hypothesis testing, and that's because there is actually an implicit null hypothesis in principal components analysis, which is that your covariance matrix is equal to the identity matrix. And this actually means that every neuron in the brain is uncorrelated with every other neuron in the brain. Every electrode is uncorrelated with every other electrode, and so on. So this is just a ridiculous straw man hypothesis. So that means that PCA is not really suitable for hypothesis testing. So now we get to GED, and what you'll see is that it's really just like a very small adjustment to the PCA optimization. So we just stick another matrix in the denominator here, which is R, and that is another covariance matrix. And I'll talk about how you define S and R in a moment. So here you can see the idea is that the null hypothesis now is that we are comparing two different covariance matrices. So we're testing whether the covariance in S is the same as the covariance in R. Now that is no longer a trivial null hypothesis because these are both coming from the same data set. They're just coming from different features of the same data set, different time windows or different frequency windows or different trials in the experiment. But the optimization is otherwise similar. We still want to find some vector W, a weighted combination of channels, such that that weighted combination of channels maximally separates S from R, so these two covariance matrices. And then in this case, lambda, this ratio would be the power, this multivariate S and R ratio. Okay, so this would be for a vector, but actually we can expand this to an entire matrix of solutions, and that allows us to view GED in pictures. So here is one covariance matrix that we want to maximize. This is like the reference covariance matrix. this would be a matrix W that contains all of the solutions, all the weighted combinations of channels. This is the matrix, a diagonal matrix lambda, which encodes the power ratio. Now, when you look at these two matrices, S and R, the first thing that pops out is that they're overall very similar, right? Obviously not identical, but they're overall very similar. And that's actually a good thing, because PCA is going to tell you what is like the biggest, most prominent features in this matrix. But the GED doesn't actually care about the most prominent features in the matrices. GED is going to find what are the patterns, which are likely to be more subtle. What are the patterns across the different channels that actually separates S from R? And in fact, anything that's common, any patterns or structure that's common between S and R is actually excluded from the analysis. Just like, if you would multiply this by like 7 divided by 7, it doesn't matter. The 7s cancel out. Okay, so I think, sort of running a little bit out of time, but I will just say that in simulation studies where we know the ground truth, GED and methods that are based on GED perform very favorably. They have very high reconstruction accuracy, even in low noise or high noise, low signal to noise ratio. And they perform much better than PCA and ICA and just picking individual electrodes. So now I'm going to tell you a little bit about how you choose these two covariance matrices. So one way to choose the two covariance matrices would be to say, you do a pre-stimulus period and a post-stimulus period. So these are laminar recordings from mouse visual cortex. Each line here corresponds to a different channel or a different depth in the visual cortex. And the idea is to find components, identify weighted combinations of channels that are maximally different for processing this visual stimulus versus just the sort of ongoing activity before the visual stimulus appears. This is a really great way to specify your hypotheses, because all of the intrinsic anatomical connections are averaged out. So you're really getting something that's very specific that allows you to test hypotheses, which is in general a nice thing to do in science. Here's data from a study about where we applied these analyses to recordings from the monkey amygdala. So this is the structure of the amygdala. Here's a picture of what the silicon probe looked like with 16 channels. So what was really remarkable to me about this study is that generalized eigendecomposition has no anatomical constraints. It has no spatial priors or anything like that. You can even totally randomize the ordering of the channels in the data. It doesn't matter for the analysis. And yet, when we look at the components that were defined purely based on the covariance matrix, we see that they mapped onto the microstructural anatomy really, really well. So you see that these components, this is a projection of these components, and they just corresponded really beautifully to the detailed circuitry of the amygdala, even though this was not imposed, the spatial information was not imposed by us on the analysis. That's just blindly what the analysis discovered, which is pretty convincing, I think. Yeah, you can also define the time windows for the two covariance matrices based on different windows of time relative to a slow oscillation. This would be an example of using GED for cross-frequency coupling, which is a particular kind of dynamic that people get pretty excited about in neuroscience. Here in this paper, we applied GED to a mix of field potential electrodes and also spikes. So you can mix totally different kinds of data as long as the data channels are suitably normalized. You can mix action potentials and LFP channels and also EEG all into the same data matrix. Again, the generalized eigendecomposition will just figure out what correlates with what and how to combine all the information together to form multiple components across different frequency bands. So that was also a pretty interesting approach. The last thing I want to show off a little bit here is an adaptation of generalized eigendecomposition for identifying empirical frequency boundaries. And so here, the idea is in neuroscience, there are, or in electrophysiology, there are different frequencies that we are interested in, right? So I was talking a lot about theta. But how do you define the boundaries for these frequencies? Usually people do it based on integer values, like 4 to 8 is typical for theta, or 8 to 12 for alpha. But sometimes people also specify alpha as like 8 to 13 or 7 to 12 hertz. So there's some variability, and there's no reason why the brain would say, okay, these frequencies should start at exactly 8.0000 hertz. So what we did here was adapt generalized eigendecomposition to identify empirical frequency boundaries based on spatial-temporal similarities in spatial-temporal structure across the different frequencies. So this you see here. So this is a correlation matrix of all of the eigenvectors from the GED analysis run on specific frequencies. And they just block off really beautifully in this correlation matrix. And so each of these blocks corresponds to a range of frequencies that is empirically determined, where the spatial-temporal structure is very similar to each other and also very dissimilar to other frequencies. It also works great in simulated data, but that's not so surprising. But here you see another example of empirical data. So we also get some what looks like artifacts in here. These are probably also artifacts. So it is important to inspect your data carefully, because these kinds of multivariate decompositions don't really know what is important and what is not important. They don't know what is signal and what is noise. All they know is that there are patterns distributed across all the different channels in the data, and those patterns are important somehow. They're different from the reference covariance matrix. Okay, so I hope I didn't overwhelm you too much. Generalized eigendecomposition is actually it's just a general mathematical background, and there's many analyses, many multivariate analyses that can be built on top of generalized eigendecomposition, including Fisher linear discriminant analysis, if you've heard of that. Or if you've taken a machine learning course, then you might be familiar with linear discriminant analysis. That's actually just, that has generalized eigendecomposition at its core. So there's a really long history of applying generalized eigendecomposition to solve multivariate problems, going back basically a century to Fisher. But I still think it's a really underutilized method in neuroscience. It's very flexible, very powerful, very accurate. And I think there's a lot more that could be done with it. So how can you do GED if you're interested? I would say the most important thing, which is the most important thing in most of science, is to have a good hypothesis. If you don't have a good hypothesis, it's going to be very hard to specify these two covariance matrices. So you do need to be clear about having an hypothesis. Then the second step is to create two covariance matrices. And again, one covariance matrix is the data feature that you want to maximize. And the other is like a reference feature that provides a sort of control condition for the S covariance matrix. From there, it's kind of simple. It's like easy in the sense that you can do it all in one line of code. I'm using a lot of apology quotes here because, of course, that the MATLAB code just to run the GED is very simple, and to compute the component time series is also simple. You just multiply the eigenvector by the data matrix. But of course, there's a lot of stuff that goes before this line of code, and there's a lot of stuff that comes after this line of code. So so it's not quite this simple, but it is fairly computationally tractable. I guess the main thing is that GED does have some metaparameters and choices that you have to make, and some tricks and tips, and so there's a bit of troubleshooting when things go wrong, and knowing a little bit about how to average and sort of avoiding overfitting and deal with inferential statistics and so on. So it does take a little bit of experience. But if you're really interested in doing this, feel free to reach out and ask me for help. I'm always happy to chat about this kind of stuff and help out. I can also recommend two general papers. This one is something that I put on arXiv a few months ago. It's currently under review. This one is also a really great paper, which you can also find on arXiv from a couple years ago. This one is, so my tutorial paper, I tried to make a little bit more focused. And this one is a little bit more general here, but they do go into the math in a little bit more detail than I do. So if you're interested in that. Yeah, they also talk about, it's just also a good paper in general. So let me quickly summarize. So midfrontal theta is an idiosyncratic signature of near mistakes. We still have absolutely no clue whatsoever where this thing comes from and what it means, and what kinds of computations are actually underlying midfrontal theta. But I am optimistic that we can make some nice discoveries about the computations and the implementations of those computations based on modern neuroscience techniques. There's a lot of kinds of questions that were just totally unthinkable, 15 years ago that are possible now, though still difficult. Yeah, and I think I'll close with this one, that proper data analysis methods are really crucial for the advancement of science and interpretation. And one example, the key example, which I kept coming back to in this talk, is my initial belief that that midfrontal theta was unitary because it looks very idiosyncratic. But I think what was really happening is that there's multiple midfrontal thetas, and they're all projecting to the same, roughly the same topographical region. So if you just look at the scalp, if you just do univariate analyses, you're just seeing how all these midfrontal theta components mix together. And in fact, you need some more detailed methods to discover that there are independent ones. Well, it was already mentioned before. I have some books and courses for like educational stuff. Finally, here is my team and my funding. I don't do all of this work on my own. Unfortunately, one day I'm going to build a cloning device and I'll fire all these people. But in the meantime, I have a collection of great PhD students and postdocs and technician, and they're all great. So thank you again, Siobhan, and everyone else for inviting me, and thank everyone in the audience for participating. I'm happy to answer your questions if you have any.
Thank you so much, Mike. This was a very, very cool presentation. I will now hand over to Siobhan for our Q&A. Thanks, Siobhan.
Okay. Super. I'm going to start off with my very simple question. Hopefully, Joaquin will come on screen to ask his. But I was curious about the task that you had the mice performing. I didn't quite hear the one similar to the Stroop test, and then also just the recordings that you were doing.
Yeah, so that was here. Yeah, so it is a rat variant of a Simon task. And we did not develop this task. It was already published in literature, so we just we took from existing literature. So the short version of the story is the rat nose pokes into a central nose poke, and then there's two sounds. That's either a low pitch or high pitch frequency. So if it's a low pitch, the rat has to nose pick to the left, and if it's high pitched, he has to nose poke to the right. Now it gets complicated when we play the high pitched tone from the right. So there's an auditory cue that's coming from the right-hand side. So that elicits this automatic orienting response to the right. So the animal wants to look over to the origin of the sound, but then they have to actually go to the left in order to get the reward. So that is the key conflict trial. Yeah, and then the recording looks something like this. So we had 30 channels implanted into the prefrontal cortex, and they were layered like this to get to try and sample as much as possible from the prefrontal cortex.
Oh, thank you very much. We have Joaquin on screen. Joaquin, would you mind letting us know where you're joining from, and then you please launch into your questions?
Uh-oh, I think you're on mute.
Quite hear you.
Don't hear you. Sorry, Joaquin.
It seems like you are unmuted on the platform, so it might be your connection of your audio.
Sometimes if you have multiple mics on your computer, sometimes the wrong mic is selected.
Yeah, or if you have multiple screens, sometimes it's feeding through the wrong screen.
I can quickly re-invite you if you would like.
Yeah, I think let's do that.
Yeah, I'm going to try that. Okay. Siobhan, maybe in the meantime you can ask Liane's question.
Okay, sure. So we have Liane's question, who thanks you for the great talk. Her question is, since you mentioned proper normalization is important, what should be taken into account to normalize data from different scales? For example, LFP, APs, and behavior. And can discretized data be used as an input as well?
I'm sorry, what was the second question?
The second question was, can discretized data be used as an input as well?
Oh, right. Okay, so two great questions. The normalization of each data channel is actually not super tricky or weird. You can basically just Z normalize every channel. The issue is that if you have one channel that is in the scale of millions and another channel that is in the scale of 0.1, then basically the analysis is never going to discover that the smaller channel is important. So the data channels need to be in the same scale. As for discrete variables, yeah, so spikes, so these are the spike channels that you see here. So spikes are actually binary when you look at the spike times, right? So it's a vector of all zeros and then it's 1 where you have the spike. So what we did here was to smooth the spike time series out by convolving it with a Gaussian with I think it was 30 milliseconds full width at F maximum, but it's basically just a way to smooth it out. Otherwise, you can have two spikes that differ by only 1 millisecond, and then the covariance between them is still 0 because they're orthogonal. So you just want to smooth out your discrete variables a bit so that they have the ability to overlap. Yeah.
Super. And I think there is sound coming from Joaquin. So
yes.
Please go ahead.
Can you hear me now?
Yeah.
Okay, thank you. Sorry about that.
Yeah, no worries.
I'm from Chile, Santiago, and I'm a PhD student in neuroscience, and I want just to thank Mike for his book. His books, they're very good, material to learn about this, and they have helped a lot of students in my neuroscience program as well. So,
thanks.
I had two questions. First, so would you recommend this GED method to study like midfrontal theta or sensor source synchronization using only EEG, scalp EEG? And any recommendations, particular recommendations?
Yeah, well, obviously I do recommend using GED to study midfrontal theta because that's what I did. I think, I think in general, I think that multivariate analyses are important, and I think multivariate analyses are better. They have more sensitivity to detecting potentially subtle effects and overlapping effects, which get mixed together at the channel level. Whether to use GED or any of these other methods, I think depends a little bit on your goal. I'm a big fan of GED, but I certainly wouldn't say that it's optimal in all cases or it's always the right thing to do. One major difference is with like decoding and deep learning. Essentially, you're just trying to predict a categorical outcome, which is great for something like engineering. It's amazing for computer vision. It's great for BCI. If you're approaching this from like a scientific perspective, you're not really just interested in seeing whether we can categorize condition A or condition B. You want to know what's the mechanism, what's the circuit doing, what do the data look like that make that prediction. And so that is harder to do with something like even linear decoding, because you usually binarize the data. And deep learning just has too many nonlinearities to be able to really make a clean physiological interpretation, which doesn't mean you can't. It just means it's a little bit more involved. So yeah, so I do recommend GED. Oh yeah, you also asked about source imaging like beamforming or dipole fitting. Those are also great. Yeah, so I mean, beamforming is also a really powerful method, also multivariate. I think one, a key difference between beamforming and GED is that with beamforming, you are doing the analysis purely based on spatial anatomical constraints, right? And with the GED, there's no spatial or anatomical constraints. It's purely statistical. So what is the goal of the analysis? If you want to see, is this, do we see conflict-related data in parietal cortex for a spatial conflict task, then GED is not the right analysis, because your question is fundamentally about the spatial organization. So yeah, so yeah, does that answer your question? Okay, thanks.
I'm not sure if, sorry about, can I do it right now, or should I wait the turn around? Okay.
No, please ask your second question.
Okay, thank you. So I'm working right now in doing a learning task, arbitrary associative learning. So I'm wondering what are the consequences of a neural perturbation and a cognitive state such as conflict and error monitoring in consecutive trials. Could you expand a bit on that subject, please?
Yeah. So in conflict tasks, I mean, I think in general, these trial sequence effects are probably bigger and more meaningful than most people look at, right? Most people just sort of ignore the sequence effects. In the cognitive control literature, we often look at trial sequence effects because of this guy, Gabriele Gatton, in the 80s, who discovered that these are really important. It also depends a little bit on the task. There are some kinds of tasks, like a flanker's task, where the trial sequence effects are modulating conflict and error processing, but not really that much. So it's kind of a relatively subtle modulation. But then there's other tasks like the Simon task, where you get completely flipped results. So the sequence effects are huge, even leading to seemingly opposite results. So yeah, so I don't know what to tell you. I think if you want to dig deeper into the data, then yeah, sequence effects are really great to look at. I don't know if they are necessarily always really crucial to look at, in the sense that, how often would you come to a completely different conclusion about the data by looking at sequence effects versus ignoring sequence effects? And there are, I would guess, for some kinds of cognitive processes, for some kinds of experiments, you would, and some kinds, some types of operations you wouldn't. I think it also depends on how closely the trials are spaced together. So Tobias Egner has a nice paper about this, looking at sequence effects as a function of the intertrial interval. And basically what he shows is that the sequence effects are really large for really short ITIs, right? Because the trials are just sort of rapid fire. But then you extend the trials, or the intertrial interval, up to, like 5 seconds or 7 or 10 seconds. And then the sequence effects are much smaller. So it does kind of seem like there's this sort of like neural or cognitive trace that's like a leaky trace that's sort of decaying over time. And so if you wait long enough, then the sequence effects get smaller or maybe even disappear.
Do you have any follow-up questions, Joaquin? Or
No, but I'm okay. Thank you. Thank you very much for inviting me.
Yeah, thank you for
Thank you,
joining us
Mike.
online. We do have a couple more questions. I'm just going to give Juliana a second to invite Sina on screen.
Okay.
Cool. Yeah, the invitation has been sent and they are connecting.
Oh, fantastic.
Hi there, guys.
Hey.
Thanks for the talk, Mike. I just so I had a similar kind of a problem that GED is solving. And my first thought was to actually eigendecompose S minus R instead of S over R. Do you have any thoughts on why one would prefer S over R?
Yeah. So basically, you really want to get this ratio in here. The thing is, so if you would do S minus R, then that's actually just a regular eigendecomposition on another matrix, S minus R. And the eigenvectors of, I'd have to think of this one for a bit, but I think the same eigenvectors that would decompose S would also decompose S minus R. So that's actually going to be pretty comparable to a PCA. There's another side of this whole story, which is that one of the limitations of principal components analysis is that the eigenvectors are constrained to be orthogonal, right? So they all have to be zero correlated with each other. But that is an unfortunate constraint for neuroscience data, because we don't expect components in the brain to be orthogonal, right? So like, you think about visual processing and attention selection and motor output. These are not orthogonal processes. They're separate from each other, but they're all highly correlated with each other. So if you're constrained to have the components be orthogonal to each other in the channel space, then you're not going to get a clean separation of sources. You're just going to be basically reducing the dimensionality to whatever is the strongest source of variance. And so S minus R is essentially just this on a different matrix. And then, yeah, the S minus R is still symmetric, so you're still going to get orthogonal eigenvectors.
Yeah, I would just wonder, like, what, for example, the primary eigenvector in that situation, how that would compare to the first eigenvector in the GED situation. Would they be very different under what circumstances, etc.?
Yeah, I'd have to think about it a little bit more, but I don't think they would really be comparable. I think those are going to be two very different kinds of things. Yeah.
Cool. Thanks.
Yeah.
Thank you. Thanks for joining us online as well.
No worries.
But wait, okay, a bit late. Just like to say, this is the final call for questions while we wait for Roderick to join us online. Otherwise, I'll ask us for Roderick.
Yeah, just give me a moment. I'm seeing now if he's coming online. Yeah, he's connecting.
Yeah, can you hear me?
Yep.
Yes.
Okay. Yeah, I was just curious. You mentioned, sorry. Yeah. You mentioned, Mike, that MATLAB rather than Python for doing GED analysis. I'm just curious if that's a packages issue or is it just particular calculations? What's the differences?
Yeah, I know. I'm always going to get flack for saying something like this.
Well, I like both, so I'm just curious what the difference is.
Yeah. No, I mean, I also like Python. I think it has, but I mean, there is like this whole bizarre culture around Python that doesn't exist for MATLAB. That these people who are like hardcore in the Python cult just sort of they take personal offense to even the idea that Python isn't the best language for everything in the world. Right? So I'm always you're gonna offend someone if you say something like that. Anyway, Python is okay. The thing is, I think there's two reasons for this answer. One is really just sort of community based, that not the entire neuroscience community, of course, but the overwhelming majority of electrophysiologists in neuroscience use MATLAB. So therefore, I think it's good to use MATLAB just because it facilitates sharing code and things like that. Then there is another reason, which is that the generalized eigendecomposition is a pretty numerically tricky procedure to run. If you're working in like toy examples, or simulated data, it's a pretty easy thing to do. But in real data, when you have large matrices, this is actually this analysis can become a little bit numerically unstable. I've tried testing, exactly the same matrices in MATLAB and Python, and MATLAB will give good solutions, and Python either just can't give a solution, or it gives solutions that are much uglier. They have sort of complex valued solutions and higher spatial frequency. And I'm not totally sure why that is, because I believe NumPy is still using LAPACK for the low-level implementation stuff, which is also the same Fortran library that MATLAB uses. So I don't know the whole reason, but I think somewhere inside MATLAB, it's just much more active and intelligent development for solving difficult numerical problems. And I think Python is just lagging a little bit in that. So yeah, but that's I don't actually have strong feelings. If someone wants to do this in Python, go for it. Yeah, it's great. Yeah.
All right. Thank you.
Yeah.
Thanks. I think that wraps up our Q&A session. Thank you very much. It's very informative.
Thanks.
Julianne, would you like to round us up?
Yes, I'm sharing my slides now. It looks like they're up on the screen. Yeah, thanks so much, Mike, for a wonderful presentation, and thank you so much for joining us online. Then to everyone in the audience, please just give Mike a virtual round of applause to show our appreciation. And then to all of you in the audience, do subscribe to NERV's mailing list and calendar so you can follow us on when our next events are coming up and who will be our following speakers. And Siobhan will be sharing those links in the comments now. And yeah, thanks to all of you for attending. This was another great NERV. So thank you so much, everyone. Good night.
Thanks, everyone. I also just put myself on the mailing list, by the way.
Oh, wonderful.
Oh, fantastic.
Yeah. Thank you so much.
Yeah.
Thank you. Bye.
Cheers.
Bye.